It's a great pleasure to welcome Josh Colston today. He's an epidemiologist and spatial demographer and works on infectious diseases, childhood undernutrition, and their socioeconomic and environmental determinants of health. What is really fascinating is what he's going to talk about, the Planetary Child Health and Enteric Observatory, PlanEO. So I looked up the pronunciation on your website. This seems really fascinating effort, which characterizes environmental drivers of enteric diseases using really interesting data sets which are put together. So we're really looking forward to your talk, Josh.
Great. Thank you, Anil. Thank you, everyone. Thanks for tuning in today. I'm Josh Colston. I'm here in the School of Medicine, mostly working in research, but I do a bit of teaching epidemiology as well. And yes, I am more of an epidemiologist in the traditional vein, a classically trained epidemiologist, not so much on the computational side yet, though I hope to change that and branch out. So what I'm presenting today is not so much results of an epidemiological analysis, though there will be some. It's more of a bioinformatic resource that we are compiling that I think will be of interest to this group, and people will see the potential and particularly expertise represented in this group. You might be able to think of some applications or novel ways of analyzing the data I'm going to show you. So as Anil mentioned, this project is called PlanEO. So we're sort of in the first year of a 5-year grant to fund this to get it off the ground. Although it does build on some work that I've been doing previously funded by NASA, in which I collaborated with climatologists at Johns Hopkins and also here at University of Virginia. So some of this work started as my PhD thesis and then my postdoctoral work, and now I'm sort of almost full-time on it. So just to explain the name PlanEO, so planetary refers to the planetary health paradigm, which you might be familiar with. I'll introduce it a little bit more later. Planetary child health and Enterics. Enterics is an increasingly used shorthand for enteric pathogens or enteropathogens. These are bugs that infect the gut. Anything with entero in the name, like gastroenteritis, means to do with the gut. So bugs that infect the guts, causing diarrheal disease. And then an observatory, because you'll see, we kind of think of ourselves as sort of observing the whole globe. And the word planeo actually in Spanish kind of has two meanings. It can mean I plan. And then there's this less common meaning, planeo, which means a sort of a gliding, which I thought was appropriate because we use a bit of data derived from Earth observations and satellites. So you can imagine this soaring satellite gliding over, observing the world, and that providing information that will feed into our analyses. Okay, so a bit of background here. So I'm introducing just the concept of diarrheal disease, perhaps not a pleasant thing to talk about. I'll try to keep it not too graphic, but enterics is kind of used as a euphemism, but we are talking about diarrheal disease, which continues to have the potential to be fatal and still causes deaths, particularly in vulnerable groups like children. So what I've got here this is from the Our World in Data website, which they do some great visualizations of this stuff. So you've got the causes of under 5 deaths over time since 1990. It's a time series, and you can see the nice encouraging trend of everything going down. Lower respiratory infection is still the main cause, but you can't quite make out diarrheal diseases here. So if I pull it out a little bit, what you see now is diarrheal disease has really been one of the most precipitously declining causes of death in children over that period, and that's something that's mirrored across all age groups. It's actually overtaken neonatal asphyxia and other neonatal preterm conditions as a cause of death due to a number of factors, chiefly the wide availability of vaccine for rotavirus, one of the major causes of diarrheal disease, and also this discovery that there's a very cheap way to treat diarrhea in children, and that's to give them oral rehydration solution, mixture of water and salt and sugar. It's available in most places and can be a frontline treatment for moderate to severe cases. And then this is just another way of looking at it. If you rank the child under 5 deaths, it's at around 500,000 per year. And, as the low hanging fruit get picked of the gains in this, that's sort of plateauing. And we're getting down to the more stubborn cases. Another way to look at it here in a pie chart, diarrheal disease is this segment of the pie, the yellow being neonatal and the other being post-neonatal. Here in this study from Johns Hopkins, I believe, they do indeed confirm that diarrheal disease has been the disease that has contributed the most gains to the decline in mortality. So it declines in diarrhea make up 6% or something of the declines in mortality since 2000. So a lot of encouraging signs there. Some more ominous things on the horizon. Diarrheal disease is climate sensitive. We're experiencing climate change. Like I said, the low hanging fruit is starting to run out. It's starting to have been picked. So we're down to the more stubborn populations that are hardest to reach with interventions. Complicating the picture a little more is that diarrheal disease is the name of a syndrome, in the same way that pneumonia is, or meningitis, that can be caused by a variety of different pathogens, whereas the symptoms can be outwardly similar, for the most part, except for a very well-trained physician might be able to tell them apart. Mostly gives the same symptoms, but can be different bugs in the gut causing the actual symptoms. So what I've done here is I've ordered the etiologies, so the bugs that cause the disease. This is morbidity, so illness due to diarrhea. I've ordered it first by viruses, bacteria, protozoa, and then secondly by the relative contribution. And you can see the viruses are in the shades of blue there, the bacteria are in the shades of brown, and then the protozoas are in the shades of green. So, we're starting to get a little bit more of a sense of the relative burden of each pathogen. And that's important because vaccines target individual pathogens. They don't target syndromes so much. And you can see that despite there being a rotavirus vaccine, rotavirus is still the second biggest viral cause of morbidity. And I believe by far the biggest cause of mortality. This second one, Our World in Data, they did a similar visualization to what I've been using, and they've ranked it as for etiology of death rather than illness. And rotavirus, in spite of our very good vaccine, is still the major contributor. So this is a picture of a child who is receiving oral rehydration therapy for diarrheal disease. Again, that's one of the most successful interventions in this space. So moving from talking about diarrheal disease to enteric infectious diseases. Some enteropathogens don't cause diarrhea. Some diarrheal diseases are not infectious. So let's talk about enteric infectious diseases. Like I said, around 500,000 deaths in children under 5. We treat the under 5 population as the kind of sentinel population because they're most vulnerable, most exposed. They're immunologically naive to these pathogens. They're most exposed because, they're crawling around on the floor, putting things in their mouths, that sort of thing, which is all, as we'll see, is all things that are risk factors for contracting these pathogens. So really, there are dozens of these bugs, dozens of different species that you can tease out, and each one kind of has its own sensitivities to the environment. Some of them will be more prone to waterborne transmission, or some of them will be more responsive to increases in temperature. And this complicates the picture, but also we're living in an unfortunate time where we have differential diagnostics, but also we have big availability of climatological data, and we can look at these specific pathogens on the one hand, specific environmental parameters on the other, and sort of see these pairwise relationships. So Planetary Health, as I was mentioning, this is a transdisciplinary approach, emphasizing the interdependence of human health with that of the Earth's natural systems. That is becoming a sort of lens by which people are viewing a lot of health problems these days, and linking it to some of these alarming processes that we see in the news every day going on: climate change, deforestation, and encroachment of humans into wild habitats, that sort of thing. But then this has coincided with really a revolution in big data and global health, and it's also coincided with a global pandemic, all of which has sort of made apparent the public appetite and the appetite among stakeholders and decision makers for data, epidemiological data in formats and in repositories and interfaces that are navigable and can be used and democratized. So the ones I've highlighted here are the Malaria Atlas Project, Johns Hopkins COVID-19 dashboard. Some of the people involved in that are also on this project. IHME does a lot of work in this space, and the demographic and health surveys, and WorldPop is a source of demographic data. These are the sort of players that we're comparing ourselves to. These are the established brands in the space in which Planeo is an upstart. If you're familiar with these, it might give you an idea of what we're aiming for. So Planeo, yes. The objectives are to provide an evidence base for targeting enteric infectious disease specific child health intervention. So that might be a vaccine, a novel vaccine. It could be other things like livestock management. Some of these pathogens are zoonotic and transmitted from livestock, particularly on a small scale. We take a big data approach to modeling these diseases. We compile epidemiological data based on PCR diagnostics, which these days everyone is familiar with the acronym PCR. And we use Earth observation derived environmental and climate data, as well as data on household level factors and subject level factors that are taken from household surveys like the demographic and health surveys and the multiple indicator cluster surveys. We do statistical analyses, georeferencing, and the plan is to make the results and the findings available through an interactive web-based dashboard and foster an international consortium of investigators, including the investigators who could kindly contribute their epidemiological data to us, as well as experts from other disciplines. And here on the right, you can see a conceptual diagram of the relationship which we think is increasingly being seen as central to understanding enteric infectious disease risk. And it's one that emphasizes atmospheric conditions, the hydrological cycle, topography, whereas household level factors have tended to be the focus previously, things like sanitation, water source. The household has been seen as the unit of risk. And we think that the conceptual framework extends beyond that to things like community-wide drainage and things like that. I'll explain more of that later. These are some photos that I took of a field site that I do a bit of work in. It's in Peru. It's in a place called Iquitos in Peru, which is in the Amazon basin. So it has a very high water table under the ground, being in a big river basin, making it very difficult to provide modern sanitation. Have some big sewers or even pit latrines, things like that. You dig down a couple of feet and you're already filling up with water, and that gets contaminated and spreads elsewhere. So it's a challenging place where you can see many of the risk factors that we try to quantify. So here you can see this is a latrine with a pipe going out the back, and basically all the effluvia is going into a little gully there. And eventually that's going to wash down into the river or into a source of water. Here you can see something that's quite common in peri-urban and rural areas in low and middle income countries, and that's a wastewater canal running alongside a street, sort of all-purpose drainage, rainwater runoff, as well as sanitation runoff, to put it euphemistically, all mixing together, easily overwhelmed when there's a major rainfall event or even a flood, and the local population very exposed there. This is a child that I met at the study site. As you can see, she looks very healthy and happy. She's nice and plump, so she's nutritionally equipped to fight off enteric infections, but she's exposed to some risk factors. This is a tropical zone, so they make do with housing constructed out of natural materials. The floors are bare earth, which is the traditional way of making a house there, but bare earth, unfortunately, provides a habitat for a lot of pathogens. So she'll have that as a sort of risk factor. She might be playing around on the earth floor and more likely to contract a bug of some sort. And then here we have some sort of chicken, I guess it is. Yeah. Type of chicken I'm not familiar with, but in this part of Peru and in a lot of places, livestock husbandry is a big source of income. It might be secondary or primary source of income. It's just one of the ways that people manage their balance of income and outgoings. And birds in particular can be a source of Campylobacter infection, but more generally, there are a lot of bugs that are carried in the poop of animals that get mixed up with earth and end up, unfortunately, getting consumed by kids. So that gives you an idea of how kind of multifaceted the conceptual framework that we're dealing with is. Environmental factors, and then household factors, and then subject factors like nutrition, feeding status. things like that. Genetic factors, we don't deal with genetic factors yet, but maybe in the future. And so just to describe the epidemiological data that goes into Planetary. So we identify studies in the literature. This is an approach called independent participant data meta-analysis. We identify studies in the literature that are recently published where they've recruited kids, used samples of stool and tested for multiple pathogens using these new diagnostic technologies, PCR-based, that are increasingly being used and which UVA has been something of a pioneer of, actually. Carried out in low middle income countries in under fives. We contact the investigators and we request access to their data. So this is sensitive individual level data. We have to set up data agreements. We have to have a data management plan, a secure place to store the data on the UVA School of Medicine servers, that sort of thing. Eventually, after all the paperwork and negotiations done, they transfer their data to us and we put it in a standardized format into a central epidemiological database in a standardized format. So we have 28 studies carried out in 25 countries so far. You can see this map's a little bit out of date, but this is showing prevalence of Shigella, which is a bacteria, as well as it's showing the number of samples and the locations. And so each sample is georeferenced either to the household, if it's known, place where the subject lives, or more frequently to like the centroid of their community or their neighborhood, or to the health facility where they were identified and recruited. And so I won't go into the details of the process flow, but we georeference these. So these are some illustrative household locations. They're not real. It's just to give you a sense of how we do it. So, if you imagine each of those yellow points on that satellite image there represents a house where a subject was tested for pathogens, then if we have environmental data about that area, we can extract it at those locations. And we can get a whole kind of suite of covariates and predictors together into this database. And that can feed into a model to get predictions. So we have about 86,000 data from about 86,000 stool samples now that are georeferenced to about 15,000 unique locations. So the type of covariate data that we're using, so remember that I talked about finished as opposed to bare earth floors. So this is a sort of side project that we have feeding into Planetary, where we're taking household survey data, these nationally representative surveys where they ask, what's your house made out of? And we classify that as finished or unfinished floors. We're also doing it with walls and roofs. And so here we have predictions for a model of the coverage of finished floors in low and middle income countries. So the browns are the areas where most people are using earth floors, and then the blues and purples are places where most people have tiles down there or something like that. We're doing this as well for water and sanitation access, household crowding, things that are more household and subject level. And as I said, that's a separate project that we got funding for, and it's feeding into this because each of these can then be used as a predictor. The next level out in the sort of conceptual framework, static environmental, these are environmental factors that just don't vary day to day, or the way that data is formatted, they're static, they're from a single time point. This is enhanced vegetation index. This is informed by Earth observation, radiation coming off the Earth that's a certain green frequency or something. And it's a metric of how lush the vegetation is, how lush the greenery is in a particular location. That has been found to be associated with various infectious diseases, including all-cause diarrheal disease. So we're feeding that into models for particular pathogens. But we have maybe like 12, 15 of this type of covariate that obviously we're able to extract for any location and then predict for any location that we have it. Then the third type, time varying hydrometeorological variables. So meteorological, meaning the weather, and then some things that are not quite weather, like soil moisture or surface runoff, that are more hydrometeorological, captures all those. And we use a model called GLDAS that has about a 25-kilometer resolution. So it's quite coarse compared to the ones we just looked at, but its advantage is its temporal resolution. So it gives 3 hourly estimates, which we aggregate up into daily estimates. So if, as we do, we know the location that a stool sample was collected, we know what bugs was present in that stool sample, we know certain factors about the subject, and we know the date on which it was collected, we can match that spatiotemporally with what the weather conditions were like on that day in that place. And then we lag it a little bit because there's an incubation period and then there's a reporting delay. But the time varying aspect of this is really something that makes it unique and has only really been made possible through our collaborations. Me as an epidemiologist, this is all new to me, but we have great collaborations with climatologists and hydrologists for whom all this stuff is their bread and butter. So some preliminary findings then. As I'd mentioned before, there's this bacteria called Shigella, which is one of the causes of diarrheal disease. And it's likely to be the next one for which a vaccine will be available. It's kind of the one that's furthest along in the pipeline. They're already getting ready for phase 3 trials and things. So we picked that as our first pathogen. We got this published earlier this year in the Lancet Global Health. This is my colleague Hamada Badr, who's expert data scientist and climatologist, was lead author. And this was the kind of key takeaway figure, which is the predictions. We did it for 3 syndrome statuses. So A is asymptomatic, B is uncomplicated diarrhea, and C is diarrheal disease for which healthcare was sought at a facility. And then, so just zooming in on this one, the asymptomatic one, this was the results of the model. We put it all into all these predictors, we put them into a Bayesian modeling framework. And this was kind of the predictions that it came up with, but this is averaged over a year. So this is kind of time static. But here in the Guyana Shield, there's some sort of hotspots. Definitely here, just sort of to the north of the equator in sub-Saharan Africa, there's some big hotspots. Madagascar and Mozambique. Cambodia, there's a conspicuous hotspot. These are fairly in line with what people know about the epidemiology of Shigella. It's very sensitive to heat and moisture and all those things that we're able to capture. So, this could be used to target a particular population for a vaccine trial if they're at particular risk. But then the nice thing is that because of the time varying nature of this, we can also construct a time series of these predictions. So you can see that in the map there, we've strung it together as an animation. Like if you imagine like each day is a frame, and so there's an animation of 365 frames. This is what it looks like. So you can see it kind of ebbing and flowing over the year. Some bits flare up and others die down. And over on the right here, we have some other seasonality metrics. So the top map is the month of maximum probability of infection or month of maximum prevalence. And it's color coded so that the mid-year is the greens and the blues, and the turn of the year is the reddish colors. And you can see a kind of hemisphere effect there, and the north having a mid-year peak. Kind of checks out with what people who are more down on the front line know about this sort of thing. B is the amplitude, the difference between the maximum and the mean of the season on average. And then we were able to pick 6 illustrative locations and extract a time series for a typical year and smooth that to show the seasonality patterns of Shigella over a typical year. Other things that we were able to do. So this is just probabilities and odds ratios for different variables and how they're associated with Shigella infection. So here on the left-hand side, you've got the hydrometeorological variables I was talking about, and you can see these kind of non-linear associations. Panel B, top left, you can see the association with air temperature is very much non-linear, and like at the very high temperatures, it actually goes down. There's an optimum temperature that's not that far off the human body temperature, at which Shigella transmissions, it seems to be like the sweet spot for that. And then we were able to identify an interaction between precipitation and soil moisture there in that surface plot that you see. And then the static variables down the other side. Panel A kind of checks out with epidemiology of Shigella over age and syndrome status. And then in panel B, the remaining variables are ranked according to their importance. So down at the bottom, the blues there, you have a cluster of household level factors that are actually quite relevant. Improved sanitation facility, floor material, caregiver education. We were able to quantify these for the first time really for a single pathogen by leveraging this big data set we were able to compile. And we hope to do this with other pathogens too, to compare this ranking, compare how different variables affect different pathogens. So Dissemination is a big component of this project. And as I mentioned, a goal of Planeo is to implement an interactive web-based dashboard. And we're working on that at the moment. We're hoping to do it here at University of Virginia, use the skills that we have here, get students involved, make it a capstone project for them, that sort of thing. We have the data science school here. We have folks up at the Biocomplexity Institute. Part of what I'm doing here is spreading the word and gathering support and buy-in from as many different disciplines as I can. And then convene an international consortium of investigators, eventually, having webinars like these, but more focused on what has been the progress in the last 6 months, what pathogen, what new data do we have, what new associations are showing up, and what new publications do we have, and that sort of thing. So here's an illustration of what the dashboard would look like. So if you're a user logging into the Planeo dashboard, let's say you're interested in a particular pathogen, you can choose it from the drop-down menu there. So let's say you're interested in Campylobacter, and you're interested in the first year of life, 0 to 11 months. You're interested in what's asymptomatic carriage of Campylobacter, what's the prevalence of that. And let's say that you don't quite trust the predictions yet. You want to see just the observed prevalence, or what's been found in particular locations. So we're going to do that. We're going to catalog that. We're kind of scraping the literature as part of another project with the WHO, where we're doing a systematic review of prevalence estimates, of etiology-specific prevalence estimates. So, for example, here, in this example, the user is seeing a bunch of pie charts similar to what we saw before, with the segment of the pie corresponding to the prevalence, the size of the pie corresponding to the number of samples. And let's say that this person is interested in Angola. They've clicked on that and they can see, asymptomatic prevalence of Campylobacter was 6.25 at that location. That's interesting. What's the source of that? And then, there's a hyperlink in the window there. They can go straight to that source. So far, this is just using information from the publications themselves, unless we have permission to use the microdata for that study. It's just kind of a way of cataloging known prevalence estimates. Then let's say the user is impressed with that and they think, okay, well, I'm ready to give the predicted estimates. I'm not big on, like Bayesian modeling, but, I'm willing to take it with a grain of salt. So they can choose instead to look at the predicted prevalence of a pathogen or a risk factor. I'll get onto the risk factors later. So this person in this illustrative example has chosen Shigella. They're looking at the first year of life. And again, they have the same syndrome options: asymptomatic health facility detected or community surveillance detected. But instead, they get a smooth layer with estimates for any location that they're interested in. And eventually, this is still a prototype, but we're working on it. And, eventually they'll be able to click on a location that they're interested in and see, that's what the model predicts will be the prevalence in this place based on its environmental conditions and everything. And we only have the Shigella article so far, but eventually, that a similar bubble will pop up and they'll be able to link and go straight to the publication in which we reported the results and findings. And then, as well as pathogen layers, eventually they'll also be able to choose the risk factor layers, and that might be the earth floors that we saw earlier. And as we model more risk factors, it'll be those. Perhaps we'll have some pre-processed environmental covariates like the vegetation index I showed you earlier. And these can be downloaded as rasters, the GIS grid format that users can then import into their own GISs. It'll be publicly available data, available for download, the way that you see with some other repositories like Malaria Atlas Project that I mentioned earlier. Okay, so in conclusion, this is a child getting a rotavirus vaccine. As you can see, it's an oral vaccine, so it's a liquid that you drink. That'll probably be the platform that new vaccines come out in. So in conclusion, there is an urgent need for etiology-specific estimates. We're going beyond outward syndromes of diarrhea. We're looking at like particular bugs. Estimates of disease burden at high spatiotemporal resolution. And this is due to climate change accelerating, new vaccines coming to market. So the diagnostics of these pathogens has improved incredibly, but often etiology studies are carried out at single locations. which gives an incomplete picture. So we're attempting to bring it all together into a common format to find patterns that would not be evident at a single location. Hydrometeorological factors play a larger role in driving transmission than previously supposed. So Planeo is going to address these challenges and knowledge gaps by making generalizable disease burden estimates publicly available and accessible, and then providing input for targeting priority populations for things like vaccine trials and other interventions, laying the foundations for scenario-based projections. So it's not within the scope of the current project to do sort of future projections of disease burden, but I see that as very much like a follow-on activity, given that we can get these projections of climate and weather, we can tweak the risk factors for different development scenarios. So we can say, suppose that coverage of finished floors increases by 20% the world over. What would that mean for Shigella transmission in a scenario where temperatures have risen by 2 degrees, something like that? We can lay the foundation for that work. Okay, well, I just want to thank the team here. So Samat Swarup of the Biocomplexity Institute is involved in this, as are the names that you see there at Johns Hopkins University. And shout out to my student, Malena, who made the illustrative dashboard. She's the one making the prototype for that. And yeah, there's my contact information there as well. So, oh, and the protocol is available as a preprint. It's under review. Hopefully it's going to be published soon, or at least hopefully we'll get the comments back. So yeah, thanks for listening and thanks for tuning in. I'm happy to take questions from the audience.
Thank you very much, Josh.
Thank you.
First of all, Josh, excellent talk. And I'm very happy to see where the project has come along in these years that we've talked. 2 questions, more essentially pointers to literature perhaps, and your take on what you think. So we have a collaborator, you I'm sure know her very well, Rita Colwell, and her collaborator Antarjit Jutla. They are preparing risk maps more for cholera though. But some aspects of it seem to be nicely aligned with this. The reason I ask is because we are starting to, as a part of our project, build on top of their work, and possibly I can see a way to connect these 2 projects, without basically getting both the projects to benefit rather than messing up your project. I really want this to do well in many ways. So I just wanted to see what you thought about it. That was question 1. The second question is, I had the pleasure of meeting Gagandeep Kang back in India, and she's been a champion for these vaccines, especially rotavirus is something that she and her colleagues are given a lot of credit in terms of developing it and popularizing it in that part of the world, and it's catching on now. But I think she's now moved to Gates Foundation, as you know, as a program officer, very senior program officer. But Mamita Niuchi, who is your colleague, our colleague, works with her closely, and we have talked to her as well. And potentially there is a way to connect this to specific interests that she might have. So I just thought I'll put forward questions based on your talk, which I found really fascinating.
Thank you. Yes. No, thanks for raising those. Those are great points. Yeah, we are aware of the cholera work. Is that related to the work that's going on at the University of Florida as well? Because I know that they've got some cholera.
Yeah. So Florida has 2 or 3 different groups, but Antarjit Jutla, who is her close colleague, works with her on cholera, and she's just really done an amazing job in making sure that this problem doesn't fall through the cracks, because people think it's done, but your slides rightly show that it is not done.
They also have some collaborators at NASA, I think.
Yeah. And one of the more, we've just written a paper on migration, but the way we started off was we were trying to understand cholera risk maps in Pakistan last year after floods. Right. So we think we could actually use these weather events to produce quote unquote real-time risk maps, which she and Antarjit have done very well for cholera, but we could extend it beyond that because, and I don't know whether Henning is around, but we have been building global populations as well, like World Pops, but somewhat different. Quite aware of Andrew Tatum and his group. And perhaps that's another place we can connect. So, just many, many interesting connecting points.
Thanks. Yeah, no, that's absolutely right. So yeah, we're definitely aware of the success that's been had with predicting cholera outbreaks. And that's kind of one of the reasons that we're staying more with the endemic pathogens, the ones where if you go to any location, you're going to find them. Whereas cholera is something that's more kind of springs up in more contained epidemics and outbreaks and things. But yeah, definitely, I think there could be some cross-pollination there. I don't know the exact inputs that are being used for the cholera models, but I think it would be nice to compare them side by side and see whether we could, there could be some cross collaboration there. And then you mentioned Dr. Kang, was that it? Who's moved to Gate?
Yes.
Yeah. So she's aware of this. She's a co-author on the Shigella paper. Yes, might be time to update her on the progress of this. I'm not sure she's aware of the new sort of framework of it being a whole initiative and everything. So yeah.
Great. Yeah, and the World Pop will come back. I mean, some Earth knows it, but we'll come back and potentially show you our populations, slightly different than World Pops, but related, absolutely related concepts.
Excellent. Yeah, no, I'd be interested to learn about. Yeah.
Yeah, really nice, interesting talk, Josh. One question more like from an operational perspective. So this is something you're expecting in real time, like local governments or agencies could use, right? So the data for this would be like fairly real time. So how do you manage all that? And it probably the predictions get stale if new data comes in.
Yeah, I wouldn't really call it real time yet, unfortunately. It has a little bit of a lag. So, with anything like this, the climate data source that you use, it has to have comparable variables going back into the past historically, which is where our data is coming from, and then projected into the future. And that's kind of been a challenge for us. So we use GLDAS, which is, as I said, is fairly coarse data, but it goes back to the time frame that we're looking at, which is back to 2005. But the disadvantage is it only goes up to like 2019. So what I was showing you there was predictions for 2018. We don't yet have it at a phase where we can get the projections of the climate variables a little bit into the future and see what's coming down the pike in terms of any upticks or early warning systems, that sort of thing. Yeah, at the moment, it's really just giving us a snapshot of how things generally are in the early 2020s, let's say. But as we progress, I think we want to experiment with data sources that would allow us to go a little bit forward in time. So yeah, great question.
Thanks.
I was wondering if you could say anything about the challenges with taking survey data, aggregating it, and then making it available through the web. So, for example, the picture you showed with the yellow dots on the houses, I thought that kind of information was not something you could usually put up on the web.
Yeah, no, we're not going to put individual location data on the web. It'll be, and those points, as I mentioned, they're just ones that I put over an illustrative image. They're not real. I mean, they're someone's household location, but they're not someone who's contributed human subject data to this database. And the human subject data itself, that's the input to the model, the epidemiological data, that stays on the server here, the secure server, and it's only the predictions that we make available, which is, it's a gridded raster, and there's really not any way that you could like reverse engineer that back to a particular person. That's a key challenge to communicate, particularly to potential contributors. We have to give very, very firm assurances about what's going to happen to the individual participant data. And of course, the survey data is made available through the DHS and all that. You have to apply for it and you have to say how you're going to use it. The individual rows and columns are not going to appear anywhere outside of our servers. It's just the end result, the predictions.
Oh, thank you.
Yeah, great question. Yep. Anil's got another question.
Yeah, maybe just a follow-up to what Stephen was saying. So there is work on, like, when you have sensitive data and you want to show something about it on spatially on a map and so on. So there's work on doing that privately. Like you can support queries like within a box, how many points there are and variants of that. So maybe that's one alternative way of making such information available.
Yeah, the Demographic and Health Surveys program, they displace their data. They call it displacing. They randomly shift it within a certain distance. So it's not the true location, but it's kind of like you can quantify the error in the location.
If there are no further questions, then I guess we'll just thank Josh, our speaker, again for a wonderful talk today. And thanks everyone for coming back for our first seminar of the fall semester, and hope to see everybody again in 2 weeks on September 28th.
I see a lot of new folks who have joined, and I just want to let them know that they can be part of the mailing list and get announcement for these seminars. They are held every 2 weeks, but let Golda know, and we can add you to the mailing list so that you can decide when to join. There are a lot of interesting talks like the ones you heard today that you folks might be interested in attending.
Yes, please do. Let me know if you need to join the list. And thanks everyone for joining us today.
Thanks.
Thanks,
Bye.
everyone. Thanks, Golda, and thanks for organizing this. It's been great.
Yeah, wonderful talk, Josh. Thanks again.
Thank you. Have a good rest of your day.
Thank you.